All right, thank you, Dennis, for the introduction. Yeah, Assalamualaikum and good morning to everyone. And yeah, welcome to our NGS webinar series, RNA-Seq Deciphering Gene Expression and Regulation from Sequencing Data. So yeah, as Dennis has introduced just now, so yeah, particularly I've involved in RNA-Seq for quite some time, working and involved in publishing papers, being an author and also co-author in a few RNA-Seq publication. So for today's talk, I'll be sharing some of my knowledge related to RNA-Seq. For example, like how to, what is the consideration when you want to set up an RNA-Seq experiment. Hopefully this will benefit to everyone here today. So if you have any question related to this topic, kindly type your question in the chat box. So you can find the chat box on the toolbar up there. Right. So please write in your question, your name, and also your email. We will only select a few questions to be addressed at the end of the session, and the question that are not be selected to be addressed during this Q&A session will be answered back to you through email after today. All right. So, and also please stay tuned until the end of the session. We have some quiz where there's a gift to be won, and also there will be a special announcement to the RNAseq service offered by Apical Service. So make sure to stay tuned until the end of the session. Right. All right, so this is my presentation overview. So we'll talk about the transcriptome and RNA-Seq introduction. Next, we'll look into the important consideration for RNA-Seq experiment. Then we look into the basic RNA-Seq workflow. And next, we look into the NovoMagic. What's NovoMagic? So we'll explain what is that later. And also, finally, there's an announcement regarding the RNA-Seq service by Apical Scientific Berhad. Right. So, yeah, as an introduction, so why we study transcriptome, right? So the main question is to understand the relationship between the genome and the functioning of cells. So in a multicellular organism, nearly every cell contains the same genome. So thus the same genes. However, not every genes are transcriptionally active in every cell. So in other words, different cells show different patterns of gene expression. So these variation underlie the wide range of physical, biochemical development differences seen among various cells and tissue and may play a role in the difference between health and disease, cell development, and also reaction against stimuli. So by studying the transcriptome, researcher hopes to determine when and where these genes are turned on or off in various type of cells and tissue or even treatment. Right. So there are many ways to look into this transcriptome. So one of the approach that we can use to look into this transcriptome is by doing RNA-seq sequencing. So what is a RNA-seq sequencing? What is RNA sequencing? RNA-seq or RNA sequencing is a technique that can examine the quantity and sequence of RNA in sample using a next generation sequencing. So the keyword here is a NGS. So you can't use RNA-seq on a Sanger sequencing because we need to have a high throughput that offered by the NGS to look into at this gene expression. Right. So, but before 2007, RNA-seq just came around 2007, which after the introduction of NGS. So prior the introduction of NGS, a researcher used an old method to look into the transcriptome, such as EST library, SAGE, and microarray. So perhaps maybe most of you haven't heard word like EST library, SAGE. So this is the old technique which during my postgraduate time we used to study the transcriptome. So yeah. Uh, some of these methods rely on Sanger sequencing, so which mean require cloning and also Sanger sequencing. So when we talk about the Sanger sequencing, there's a huge investment that needed in terms of human resources and also budget. You need to have people to prepare the sample and also to prepare the library because you are having the keeping the library to be you have to keep the library in the cold storage in the -80. So all this has to be considered when you want to study transcriptome in old time. So this because of this, not many groups can do transcriptome study. So this is hinder the pace of a transcriptome study. Microwave Microarray, in other hand, may improve this. However, due to the probe-based experiment, there's a limit on the detection range. Also, the limitation on the microarray that is that the non-model and or novel organism do not have access to this platform, since microarray panel design require gene sequence information. Right. So with the introduction of the NGS and the RNA-Seq later, helps to improve on this limitation and enable researcher to tap into the transcriptome studies. So yeah, so basically the RNA-Seq starts with a sample of interest. Well, you need to do some treatment. You have to compare like whether treated or untreated sample. So next, you have to extract the total RNA. Next, from the total RNA, you can proceed with the library construction, which you need to do fragmentization, and then reverse transcribe, ligate the adapters, amplify, and once the library is ready, you can sequence using NGS platform. For example, Illumina, HiSeq, NovoSeq. There are a few other platforms available right now, like MGIT7 and so on. Yeah, but more or less the approach, they are the same, right? And next, once the sequencing process is completed, you will get millions of sequence reads, which you need to process to get the information from the sample. So the analysis require us to either do the mapping of these reads or assemble, right? And then further down the line, we can do the expression profiling, expression analysis, and so on. So there are many things that we can do with this transcriptome, this RNA-Seq data. Right. So before we go into the RNA-Seq, like result, what we can achieve for the RNA-Seq, I would like to also talk about what the most important thing before we start the experiment. So this is the things that required, the important things that is needed before you start a RNA-Seq experiment. So, for example, we look into the samples and experimental design. So we can look into the animal part. Working with animal and plant require us to harvest specific part of organism, because definitely we can't. For example, when you work with the animal model, for example, like mice, you can't use the whole mice to extract the RNA. So you need to harvest part of the organ. So the organ selection should be based on where the transcriptome profile change you would like to see. So for example, using animal model, on like mice, to study a microbe infection, we might need to focus on organ that involve in the immune response or reaction towards the infection, such as spleen or liver. All right, so that's one point. So next, like replicates. Replicates is important but always been neglected. So we always receive an inquiry from customer. They would like to send an RNA-Seq sample for RNA-Seq sequencing, but they only have one sample, no replicates. I mean, like they have two samples without replicates. It is crucial. Replicate is very crucial to improve the statistics analysis and to remove false positive on the RNA-Seq result. For and always you can see when you try to publish a paper related to the RNA-Seq experiment, the review will always request for the replicates. So it's difficult for you to publish an RNA-Seq experiment without having a replicates. All right. And talking about replicates, so it depends on the question is how many replicates needed for the experiment, for each of the experiment. So for this question, it depends on the what type of sample you're working on. If let's say the samples you're working on is in a control condition, or a subject which variation between samples are low. For example, you're working with a model organism, again using mice. Mice are being bred, I mean they are controlled bred, so you won't, the variation between one mice to another, technically they are very low. So having a minimum replicate of three can be used. I mean, can be accepted. However, like if you're working with something an organism which have a big variation between one to another, for example, like human, you're working with the clinical studies, or perhaps you're working from plant, from field, so you expect that the variation between samples are very high. So the number of replicates, the higher number of replicates will be better to have a better RSA result. Right, so some might suggest that having six and above for this type of samples, right? So that is why for the best is you have to go back to your paper, see how people work on replicates, then you can follow how they work on the RNA experiment. Right. Also, the sample collection and storage, it's very important because when we harvest the RNA, so we need to ensure that the RNA profile are locked during the sample we harvest. For example, when we collect an organ for a mice, we need to snap freeze right away rather than we leave it for, let's say, a few minutes or even few hours, because the RNA might change and it might not be reflect to the RNA profile that we actually what we are trying to see. So it's very important that sample collection and storage need to be considered when you design the experiment, right? And also setting up the sample comparison. So we need to look into when you want to do the RNA-Seq experiment, you want to compare one sample to another. So you need to carefully decide or select what kind of like sample you're looking at for or time point, if you are looking into the time point experiment, or different treatment dose. If a dose is too high, might not be showing, or if the dose is too low, you might not seeing the effect on the profile, on the RNA-seq, the transcriptome profile. If it's too high, might perhaps your animal might die, and so on. So this is need to be considered. So different type of samples, time point, what's the best time point, also different the dose, that the best dose to be introduced to the subject. Right. Also on the sequence and analysis, some of this consideration that you need to look into the what are the sequencing depths that you need. We'll talk about that during in the slide later. And then also we need to look into the analysis strategy. For example, whether your subject is the reference genome for the subject is available. So if it's not, then you need to look into how to analyze the data using a de novo approach. If the reference genome is available, then you can use a reference mapping approach. So we also touch a little bit on that in my slide. And also the last part is the validation. So this is very important. We want to publish the data, publish the result is always, you need to validate the finding from this NGS. So normally qPCR is one of the way to validate the result of your findings. Right. So let's move on. So okay, let's look into the basic RNA-seq workflow. Basically, for the basic RNA-seq workflow, I can split it into three part, which is the first, the sample preparation, library construction and sequencing, and also the bioinformatic. Right. So in the sample preparation, you need to run the experiment, and then next you have harvest the sample, and also need to store them. Right. And then look into the RNA extraction. in then in the library construction and sequencing, so we look into the sample QC. If the sample is good, then we can start with the library construction. And once the library construction is completed, then we can start with the sequencing. In the bioinformatic analysis section part, we can from the data generated by the sequencing process, we can start with the data QC, and then data analysis, and also data interpretation. Okay, all right. So for the sample preparation, I would need to very emphasize that the good total RNA quality is very crucial. So you need to have a good total RNA quality to ensure the success of the library construction and sequencing. It does affect the result on the RNA-Seq, and also the total RNA does affect the result. Right, so we look into the sample purity, which is 260, 280, and 260, 230 goes to be around 2 or close to 2 should be fine. And also one of the criteria, the RIN number. So the RIN number must be high RIN number. So RIN number tells us the RNA integrity, whether RNA started to degrade it or they are still intact, which is the RNA are still well, not degraded. Okay, so to ensure that the samples in a have a good integrity, is always have it's advisable to start with a fresh sample. uh Sometimes you might not have the ability to have a fresh samples. For example, we work with the animal model, you might need to have different what they call treatment. So you might need to store them, or perhaps time point analysis. You might need to have longer time. You can longer time to prepare the samples, so you can extract the RNA directly. So you need to ensure that you can keep the samples in the cold storage, for example, like -80, so that the RNA, or perhaps in the, for example, like in the RNA later, to lock the RNA profile. So, and also not only to lock the RNA profile, to ensure that the RNA integrity are being well kept. Right. So looking back in the read number, so using the bioanalyzer, we can have the read number from 10 to 1. So you have if you have the read number of 10, that would be the best, which is the RNA is fully intact. 8 to 9, which is a high quality. 6 to 7 considered acceptable range. While 5 and below we consider degraded. For apical scientific, and we accept RNA that is at least 6 and above to be sent for RNA-seq sequencing. Okay, right. So this is our sequencing, our RNA-seq guideline. So if you like to send the sample to us, we do accept our extraction, extracted RNA. Okay, so this is the the quality that is needed. So you might you need to have amount of as small as 400 to 800 nanogram, depends on the sample type. Okay, and the concentration must be more than 20 nanogram per microliter, with the RIN integrity number can be as low as 6.0 for prokaryote and 6.3 or 6.8 for eukaryote. And as I mentioned just now, the sample purity, the OD260, 280, and 260, 230 must be around 2, which is to show that the samples are in good quality. Right. Apart from that, some customer might have difficulties on getting a good RNA quality. For those have such difficulties, we also offer RNA extraction service, which you can send the plant or animal tissue to us, or even cell culture. We can do the RNA extraction for you. Okay. Yeah. Okay. So once we have the good RNA, so the next step is library construction and sequencing. So start with process the RNA to become a double stranded cDNA. Okay, and from there we can do the fragmentization, and then we can do the N repair, phosphorylate, A tailing. So this is general library construction. Okay, and then once we have the A tail, and then you can adapt the adapters, sequencing adapters, and once it's done, we can put this sample for sequencing. Right. So talking about sequencing depth. Okay. Right. So there are a few factors that need to be considered. The first thing is the sample type, and also the analysis purposes. All right. So for prokaryote, because when talking about the RNA-seq, unlike whole genome sequencing, we can calculate the coverage. Like normally for de novo assembly, you might need like 50 times, 30 to 50 times of coverage, so you can calculate directly based on the genome size. But for transcriptome, we can't do that, right? So it need to depends on the best case scenario. So we have a lot of publication, a lot of uh, the community have studied quite a lot and see what's the sweet spot for they say to get what's the recommendation for the sequencing depth for any type of samples and also the purposes. So for prokaryote, for example, minimum requirement is about 1.2 GB and recommended, which is a 2.4 GB. For fungus, minimum we need is about 2 GB. But if you're looking for low gene expression, you want because you need to capture the what they call the gene that express lowly expressed, or also you want to look into the genes structure analysis purposes. So normally this is if let's say these genes are lowly expressed, then you might need to increase the sequencing depth for that sample. So 12 GB, if you like to look into this, any samples that you want to look into the low gene expression or gene structure purposes. This is same goes for the animal plant. So 6 GB minimal requirement. 12 GB if you want to look into the low gene expression and gene structure analysis purposes. All right. So okay, all right. So this is on the bioinformatic part here. So once we have done the sequencing, so we need to analyze the data so that we can make sense what's contained in the data in the sample. Okay, so so if you have the reference genome, if the reference genome is available, then you can opt to use a reference mapping approach, right? So to use this reference mapping, so there are a few things which is very crucial. Reference genome, not only reference genome must be suitable. It is very important. For example, if you have reference genome, but when we try to map the reads to the reference genome, the percentage of reads mapping is like 30% or even lower than that. So it tells us that the samples and reference genome could be different organism. I mean, they are not the same. So so it must be suitable, must be correct. If you are working with mice, then you need to have a mice reference genome. um so yeah, and also you need to have the annotation available. So without the annotation, it will be difficult to look into the gene expression profiling and also diffusion gene expression. So these two things must be available. If the reference genome is not available, so there's another alternative, which is we look into the de novo assembly. So what's the de novo assembly is we construct the reference. In this case, we construct the transcript. We assemble the reads that we send for the sequencing. First, we take the reads to assemble, make it a transcript sequence. And once we have the transcript assembled, using the assembled transcript together with the reads again, we map the reads onto the transcripts, and we can continue with the downstream part analysis. Right. So basically the process they are more or less the same, reference mapping and de novo. Right. So we have the reads, and then we map the reads to genome, or for de novo assembly, we map to the transcript. So using the information, the mapping information, we can extract the expression profile of each gene, and then this has to be done separately for each samples. Right. So once this mapping and expression profile is done, then we can combine all the sample for the differential gene expression analysis. And from the outcome from the differential gene expression analysis, before that, for differential gene expression analysis, it is important to have at least two groups of samples. Of course, we need to compare, need to have two groups, and each group, it is necessary to have replicates, right? If I mentioned earlier in the slide, so need to have replicates for RNA-seq experiment, at least three, at least a minimum of three replicates for each group. Okay. And once when we do the differential gene expression analysis, we will get the list of differential expression gene list, or we call it DEG. And this DEG we can submit it to for the function analysis. Right. So this is a basic workflow for the RNA-seq analysis. So let's look into in details for this. Right. So yeah. So for this expression profiling, so from the mapping data, so we can extract the information for the expression profiling. So how does the what done was is based on this example. So we have this gene here. This is the genome with the annotation gene A, gene B, and gene C. And when we map the reads, all the reads will be mapped onto the gene. Okay, in during the expression profiling, the tools, the bioinformatic tools, can convert the mapping reads to the gene expression profile. So there are few software that can come out with this expression profile, and the outcome here will be the expression numbers like based on the FPKM. You might heard the FPKM, TPM, RPKM. So these are the unit for the gene expression profile. All right, so this is just an example. Okay, and yeah, next. So once we have the expression profiling done for all every samples, okay, then we can look into the differential gene expression analysis. So we can look into the DG cutoff to determine differently expressed gene. So normally, if you look into papers, you will always see the in the method it was stated like the what they call it the DEG cutoff log 2 for change 2 or minus 2 and pre-adjusted value of less than 0.05. So basically what does this number tells us that anything that's a positive value, for example here if it's used log 2 fortune 2 or minus 2, so 2 and higher means upregulated. Anything that's negative value minus 2 and lower they are downregulated. So this is an example of some data. So once we have this data in Excel, so if you put this threshold in the Excel, you can sort the number. So based on the log24 change and PADJ, when we sort it, so we can see anything that is minus number here. So they are down regulated. Anything that positive here, they are up regulated. So in other words, that we can see there's what are the expression profile changes from, for in this case, treated and untreated. So you can see the expression profile change. All right. Okay, so once we have the list of DEG list, so what we can do from this list of genes, from the function analysis, there are many types of enrichment and or analysis that we can do. For example, we can look into the gene ontology enrichment analysis or CAG pathway enrichment analysis, protein-protein interaction, disease ontology enrichment analysis, uh, functional annotation on oncogene and rectum enrichment analysis. So enrichment analysis is a computational biology method that identify the biology function that overrepresented from the list of genes. So in this case, the DEG list, DEG gene list, that might have an association to the different phenotype or treatment or based on what your experience set up. All right. So from here we can identify what are these biological function that might be kind of represented in this case. So this is some example. Right, so once we do the gene ontology, we might able to see what are these function that might be might highlighted from this enrichment analysis. Right. So based on my experience when running a transcriptome analysis, well, when you start with that, when you come to this point, you will have a huge amount of data. So it's easily for you to be lost when you try to dig into the data. So the first thing that I will look into when I want to make sense of what this transcriptome result looks like is I will look into this enrichment data. So in my experience, I'm working with mice infected with a microbe, a pathogen microbe. So the first thing that I will look into is the genes that involved in the immune response, because we expect that the immune response will be triggered, will be switch on. So we expect that this other gene will be highly expressed or being differently expressed in this case. So from there, we look into the pathway enrichment to see if there's any, for example, like cytokine interaction. Is there anything related to, anything related to the immune response? That will be my first pickup. So that will be, so this functional analysis will actually assist you to look into data. So from big huge data and then zoom into a very meaningful biological function information. Right. So that's why it is important that we come to the first point: the experiment setup must be well prepared, must be well done. If it's not, it's always come to the where if your experiment design are not well prepared, they always come to the point where come to the enrichment result, you won't get anything. So that's something that is have to look into again, what has been done during the sample preparation. All right. Okay, right. So apart from this, apart from the transcriptome information, like the differential gene expression analysis, uh, functional information, enrichment result, we can also look into the variant core analysis because we use the reads to map to the genome. So we can do the variant core analysis and we can see if there's any of this and since that the reads are actually mapped to the coding region, then we can look into if there's any of mutation occurs on the coding region, which is sometime it might cause the expression changes to the samples. Yeah, so that's another thing that we can do. This is something that's only available for reference mapping, not for de novo assembly, because de novo assembly is only to the transcript, which is not really kind of like I mean, it's not that meaningful if you look into the uh, variant core for de novo assembly. So normally for variant core analysis is normally will be meaningful if you start with the if this experiment, the bioinformatic analysis is using the reference mapping approach, right? So for the variant core analysis, we can look into the SNPs or indel to see whether there's any mutation that occurs in the coding region. All right. Okay, all right. Yeah. So basically, that's what we can gain from this transcriptome analysis. There's a lot of information there. Right. So just another thing. This is what to highlight. That is one thing when we do the transcriptome analysis. One of the difficulties is to select the DEG threshold. So if you see papers, some papers they might select like uh like log2 for change 2 minus 2 PADJ 0.05, but some papers they might have different threshold. So some service provider that might stick to like one threshold, and then once you get the result you can't change it. But our service is using if you subscribe to our service, we can can have access to this portal we call Novo Magic, when you can reanalyze the data. But however, this is only limited to RNA-Seq analysis with reference genome, right? So using this Novo Magic, you can revise the what they call the DEG cutoff. You can change the pre-adjusted value or the log2 for change as you required. For example, if you have a log2 for change 2 and also minus 2, and then PADJ 0.05, you get the result. It turns out the number of DEG lists to be very little, and it doesn't make any sense to your function analysis. You might want to make it a bit wider. I mean, the log2 for change want to increase to perhaps want to make it 1 minus 1. Then you can change the parameters, the DEG cutoff here, and then you can rerun again the analysis. So you will not be, you can reanalyze the result and tune to according to your needs. All right. Okay. All right. So that's the part, the final part of my presentation. Okay. So I hope that you have like some enlightenment from my talk today. So yeah. So if you have any question related to today's talk, yeah, so can feel free to write your question in the chat box and we'll address your question.